Hello, everybody. Let's settle in for conversations about cane toads and vaginas. And you make whatever connections you need to make by the end of it. My name's Laura Crouch, and it is a great pleasure to introduce Dr. Lee Ann Rollins, who is here today. She is a fellow in the School of Biological Earth and Environmental Scientists. She's also a professional photographer. And today she's going to talk about cane toads, and in particular, the thing lay people are most afraid of, genetic modification going out there. So I'm going to welcome her to the stage. She will potentially scare us, and then we'll throw to questions at the end. Thank you. Let's welcome Lee Ann.
Thanks. Should humans manipulate the genomes of other species? The truth is, humans have been manipulating the genomes of other species for more than 10,000 years. We interbreed and we selectively breed species, like our pet snoodles and the Granny Smith apples that we love to eat, and we don't think twice about it. Some of us eat genetically modified organisms, or GMOs, like canola or soy. But today, I want to take you on a journey to talk about a new gene editing technology that has the ability to affect all of us: synthetic gene drive. When GMOs first came out, the conversation was often centered around irrational fears. This time, let's be smarter, and let's use reason to guide our opinions about synthetic gene drive. Gene drive is created by the marriage of two technologies that occur independently in nature. The first is selfish DNA, which are genes that bias their own inheritance by replicating copies of themselves across the genome. And they violate rules of Mendelian inheritance. Gene drive, selfish genes were first discovered in the 1920s in Drosophila, but they were made famous in 76 by Richard Dawkins' best-selling book, The Selfish Gene. The second phenomenon that's coupled together with selfish genes to make gene drive are clustered, regularly interspaced palindromic repeats, which you guys might know as CRISPR. CRISPR was discovered in the 1990s by Francisco Mojica, and we now know that these repeats are really important to adaptive immune function in bacteria. So we find these in naturally occurring in bacterial cells. So around the same time that Francisco Mojica was discovering CRISPR, a guy named Philippe Horvath was at Strasbourg studying his PhD, and he was thinking about sauerkraut. So his project was to understand the bacteria that are used in the fermentation process. And he went on from his PhD to work for a company that makes yogurt, so I reckon this guy's a foodie. And his job there was to figure out how lactobacteria could fight off viral infections. And what he discovered was a protein that he's called CRISPR-associated protein 9, so Cas9. And this protein is essentially like molecular scissors. It cuts DNA. So if you have a look at this diagram, you can see that when a virus enters a bacterial cell, a small piece of the virus's genome gets cut and inserted into the bacteria's genome. And so what happens is when a second invasion of that same virus comes into the bacterial cell, the Cas9 complex can go and find that virus and cut its DNA. So this is a naturally occurring phenomenon. We've hijacked that to create CRISPR-Cas9. So this diagram shows you that instead of having a piece of viral DNA to cut some DNA, we can actually make our own construct. So this is synthetic DNA that gets coupled with the Cas9, and it can take that complex to a specific place in the genome. And we can cut it, and we can insert our own piece of DNA, so something that we've made into that space. So these two concepts, selfish genes and CRISPR, were put together to suggest this idea of making synthetic gene drive. So Kevin Esvelt and his colleagues at Harvard are the people that first came up with this concept that we should put these two things together. So in 2014, they published a paper where they said, you know, let's give this a try. If we put these two things together, we should be able to drive a gene, and by association, its characteristics, through an entire population in a short number of generations. So if you have a look at this diagram at the top, this is what normal Mendelian inheritance looks like. And the second diagram shows you that all the offspring now carry a modification. So that's what gene drive looks like. So the reason that this can happen is that even if only one parent carries the modified gene, all of its offspring will carry 2 copies of that modification. And this happens because it replicates itself within individuals. So this eliminates variation, and it dodges natural selection. So if we think about those genetically modified organisms that we talked about earlier, or traditional genetic manipulation, those organisms were traditionally made to improve agricultural production, and they're not very competitive if they get out into wild populations. In stark contrast, gene drive is designed specifically to be released into wild populations. So even if a mutation is deleterious to the organism, it can still be driven throughout the population because of this ability to replicate itself. So this sounds kind of scary, right? That you can actually drive something through a population in an uncontrolled fashion. Kevin Esvelt thinks it's scary too. So he's recently come out and admitted that suggesting that the use of this technology in the wild was actually quite a dangerous suggestion until we have good technology to control it. And so he and others are now working on reversal drives, which halt the action of a gene drive, and also daisy chain drives, which are basically multi-component drives that are self-limiting and will eventually succumb to natural selection. These things are really important for us to be able to control gene drive in wild populations. We really have to get this down first. So there are a few projects across the planet who are talking about actually doing this in wild populations, because it hasn't actually been done yet. Target Malaria is one of these projects, and this is a Gates-funded project in the UK, and their aim is to release gene drive in wild mosquito populations in Africa by 2029. G-BIRD, or the Genetic Biocontrol of Invasive Rodents, is a collaboration between Island Conservation, the US Department of Agriculture, CSIRO, and universities here in Australia and in the US. And their plan is to eradicate rodents using this kind of technology on islands, and this includes 6 islands in Australia. And many of you would have heard of Predator Free 2050, which is New Zealand's bid to get rid of invasive species like possums, and they've discussed the possible use of this technology. You may be sitting there wondering, why on earth would you even consider the use of this technology, given that it can spread uncontrollably in the wild? Well, if we can harness the problems associated with controlling the technology, it has the potential to solve some of our greatest problems. Every 30 seconds, a child dies of malaria. And we spend $12 billion a year to control it. Invasive species threaten biodiversity and agricultural production worldwide and cost us hundreds of billions of dollars a year. Gene Drive has the potential to solve these problems. But clearly, we can't move on to using Gene Drive in wild populations until we solve technical problems. And there are also some real risks that we have to consider. So one problem is that invasive species are all native somewhere, and they play important roles in their native ecosystems. So think about those possums again that we talked about in New Zealand, where they spread tuberculosis and they're considered pests, but in Australia they're protected. So it's really clear that if we release Gene Drive in invasive populations, we absolutely need to make sure that these changed genes can't get back into native populations. I'm an invasion biologist, and my job is to create the science that is used to manage invasive populations. Through my work, I know all too well the unintended consequences of unintended releases. And here in Australia, we know all too well all of the problems that have been associated with those invasions. So sometimes even containment facilities fail to contain, such as in 1995, when Khaleesi virus was unintentionally released from Wardang Island at a CSIRO containment facility, and it infected mainland rabbits. So many of us don't care about that, right? Because rabbits are pests too. But the problem was that this unintentional release meant that public engagement in this issue never occurred. One of the other big problems is that when we change an organism in an ecosystem, we don't actually know what cascading effects might happen in that ecosystem because they're really complex, and often a single change can cause a chain reaction, and we might not be able to predict what that is. So I want to use cane toads as an example because they're a great one. I'm sure you guys all know that cane toads were brought to Australia to control cane beetles, but instead they just powered across Australia, and because they're toxic, the Australian native predators often suffered. So species like goannas were devastated in their wake. However, the species that are typically prey of goannas, so things like children's pythons and crimson finch nestlings and turtle eggs, these things all benefited from fewer goannas. Some species of birds use cane toads as prey items now. So they've learned that they can pick up a cane toad and flip it over on its back and eat the non-toxic body parts. Since toads have invaded Australia, black snakes have evolved to have smaller heads, and some species of water pythons have fewer tapeworms because cane toads act as a terminal host for this parasite. So these are just a few things that have happened that we know about. And this has all happened in less than 85 years. So now it's really hard to predict what would happen if we actually removed cane toads from this ecosystem. In addition to ecological problems, we know that that Gene Drive has the potential to cause some really serious biosecurity and political issues. So wild animals don't respect borders, and what's good for one country may not be good for another country. Gene Drive clearly has the ability to wreak havoc with biosecurity, which is one of the reasons why the US Department of Defense is the largest funding body for this research worldwide. And also, we know that Gene Drive has got a high likelihood of giving bioterrorists a weapon, because an animal that's modified looks exactly like an animal that isn't modified. And even the introduction of a couple of animals could cause serious damage in a native range. But there are also ethical considerations. Do we have the right to eradicate a species for financial reasons? What about for human health gains? Some people might argue that we have a responsibility to care for each other first. And I don't think anyone really mourns the loss of the smallpox virus. I'm here today as a representative of the scientific community, asking you to engage with this issue because gene drive can affect all of us. Let's not let fear guide us, but rather let's take this opportunity to participate in the debate about whether, as a planet, we want to use this technology, and if so, under which terms. If there's anything here that we should fear, it's complacency. Let's not let this be another opportunity that's missed for public engagement in an issue that has the ability to affect life on Earth. Thank you.
Thank you so much. Thank you so much. I feel like that was kind of brave. So thank you again. So throw your hands up and I'm going to run around with a microphone. But the first question I just wanted to quickly ask is, what do you think that kind of public engagement that we need to have will look like? And then I'll come to you. You go.
I suppose what I would really like is that everyone in this room goes and talks to people about what you hear today. And when you see this in the paper, read it. Don't just gloss over it. Educate yourselves about this because it really does have the ability to impact all of us. So don't step back and leave this to the scientists.
Is your question?
Hi, thanks very much for the talk. I was wondering if a gene drive could evolve naturally, and if that was the case, and someone used it as a bioweapon, would there be any way to prove that it was an act of terrorism and not just an act of random chance alone?
There are natural gene drives, so I suppose it's possible that it would be difficult to trace that. But, how would you ever know? Even if it wasn't natural, how would you ever know who was responsible for it? It would be difficult, right?
There's a question over here.
Thank you very much. I was wondering about the example of feral cats, which do a huge damage in Australia to native wildlife, and what a problem that would be when there's so many people in Australia who just love their domestic cats. Is there a solution?
I personally feel like it's very risky to release technology like this into the wild because there's a lot we don't know. So, for example, with the calicivirus release, the problem with that event was not a quarantine issue. The real problem was that it was a basic misunderstanding of biology. So we thought that rabbit-to-rabbit contact transmitted calicivirus, and so they built their quarantine facility to avoid that. But in fact, flies act as vectors. We didn't know that, right? So I guess my point would be that we have similar gaps in our knowledge of gene drive, and it's a really risky thing to release it. So, some people advocate that, for example, cane toads are a good species to use it in because there are no other buffonid amphibians in Australia. But, we're really bad at containing things. So just because there aren't any here in Australia doesn't mean that it won't make its way to a place where there are some. So I would say the same thing about the cats.
There's a question over here.
Hi, thank you so much. So you mentioned the potential of using gene drive in mosquitoes. How likely do you think that is, and what are the things that are stopping that, or the risks that could come with that?
Yeah, I think that's going to happen. I don't think there's going to be much to stop that. Right now, the Target Malaria Project is doing traditional control in areas and trying to assess the type of impacts that removing that species from the ecosystem will have. I think it's quite naive to think that there would be none. But I do believe this is a freight train moving in a direction especially in a place like Africa where people they have less of a voice to argue against things. So I do think that will happen.
That's an interesting question, because I gathered that the use of a gene drive to control that mosquito actually has worked in laboratory situations, so they know it'll work.
Yes.
Now the question is whether they take it out into the bush.
Yeah, I mean, it has worked in the lab, but working in the lab and working in the environment, as you well know, is not the same thing. So right now, I actually don't think that we're tech savvy enough to actually do this today. But these are problems that will get surmounted. So I think we'll get there. And doing it in short-lived invertebrate is one thing. Trying to translate that to a vertebrate is something else completely. And I think that is actually quite a way off.
Hi, do you know of any research that has looked at gene drive in instances of hybridization?
So, I mean, obviously, if you have a closely related species, you put a gene drive into one, it could hybridize and it could move into the other, which is the whole argument about the cane toads being a good system here because they don't hybridize with anything. So absolutely, gene drive can move horizontally in that manner through hybridization.
We only have time for one more question, which is coming to you. But please remember that all of our speakers are going to be around, and seek them out and ask more questions. Here you go, last question.
If the G drive is kind of, out there, is there a future in which nobody could claim that they are GMO free?
Well, I just want to make a really tricky point here, and that is that genetic editing using gene drive and CRISPR is not genetic modification. They were very tricky when they named gene editing and avoided on purpose having an association with GMOs because of all the regulations that are around GMOs. And frankly, those GMO regulations have resulted in us, basically multinational companies are the only ones that can afford to create GMOs now because of all the regulations. So we sort of cut ourselves off at the knees. CRISPR, the terminology, you'll never hear the word modification in that terminology. So your question was about whether or not there'll be the world will be full of these things, essentially. Anything's possible, but I really hope that all of us use our brains and talk about what is appropriate to have, because without people in the general public opening their mouth about what's okay and what's not okay, it'll be completely uncontrolled. And a great example is Kevin Esfelt has been on Nantucket talking to people about rodent control there. The community said, we don't want this, and he said, that's fine. And that was the end of it. So we do have power. Thanks.
Thank you so much, Lee Ann. That was fantastic.